Hello and welcome to today's video on organelles at work. So let's get started. The synthesis of a protein illustrates the division of labour within a cell. And this is the one given by the textbook. So I'm just going to go through it. It's about making a hormone. So the instructions to make a hormone are in the DNA in the nucleus. Okay, so we learned from the last video that the nucleus is the house for all the DNA, and the instructions to make a hormone, which is also a protein, are inside. The specific instructions are on a gene, and a gene is on a chromosome. So a gene is basically just imagine it like this. There's a little bit here. This is the one that we want, so this is the one that's going to come out of the nucleus. Okay, well that's not a brilliant diagram, but you can imagine that's the small part that wants to make the protein or the hormone. So the nucleus copies the instructions in the DNA into mRNA, which we'll learn more about as the course goes on. The mRNA leaves the nucleus through the nuclear pores and attaches itself to a ribosome, and that attaches itself to the rough ER, which we learned about in the last video. The ribosomes read the instructions and assemble the hormone. As we know from the last video, ribosomes are the site of protein synthesis, and so a hormone is a protein, and so it will assemble the hormone that it's been given instructions for. The assembled protein, or the hormone, inside the rough ER is pinched off into a vesicle and transported by the Golgi apparatus, or the Golgi apparatus. And then the Golgi packages and modifies, ready for release into the vesicle, and moves to the cells, and that moves to the cell surface membrane where it is secreted. So as we learned in the last video, the Golgi modifies and packages the proteins into little vesicles so they can go and be secreted outside the cell to do some kind of hormonal process in this example here that's given by the textbook. And the next thing to learn about organelles at work, on the next thing in this textbook, is the difference between eukaryotic cells and prokaryotic cells. So basically, eukaryotics have a nucleus. I remember this by you being me, and I have nucleuses in my cells, so that's a eukaryote. And a prokaryote does not have a nucleus as such. And what else can we say? There is only one membrane, the cell surface membrane, around the outside. So it doesn't contain membrane-bound organelles that we learned about in the last video. An example of a membrane-bound organelle would be like a mitochondria. So it doesn't have any of those kinds of organelles. It has a cell wall, but it's not made out of cellulose. It is made out of peptidoglycan. You just have to kind of remember that, peptidoglycan. It's kind of a nice little word that rolls off your tongue. And that's what the cell wall of a prokaryote is normally made out of. And then how I have always described the DNA in a prokaryote is naked and homeless. That basically means that the DNA is not surrounded by a membrane, and it's kind of just loose within the prokaryote. We call the area in which the DNA lies the nucleode and the nucleoid. I'll spell that out for you. N-U-C. Oh no, I forgot. N-U-C-L-E. D. That's the general area in which the DNA is in the prokaryote. And some prokaryotes have flagella. That basically means they have a little tail to help them waft around the cell, to waft around whatever they are in actually. So they can just have a little bit more mobility wherever they are. And that's basically all you need to know for now. I'll see you again soon.